acetic acid bacteria are one of the main spoilage groups in fermented beverages, but in this context, many of their characteristics are still not well understood. My name is Saverio Castelli and I am an enologist, so very passionate about wine and about fermented beverages. This passion started a long time ago. In fact, I attended a bachelor in viticulture and enology in Italy. And after that, I moved to Bordeaux in France, where I attended a master's degree in wine science. During this master, I developed a new passion, the microbiology. This subject had already interested me a lot during my bachelor, but during my master, I had the opportunity to work at Château Margaux in a winery in Bordeaux on a project aiming to reduce sulfur dioxide dose in wines using microbiological strategies. After this master, I completed another master's degree, again in Bordeaux, focused on research in wine science. Also during this period, I spent 6 months working on a microbiology project during an Erasmus. I studied the glycerol metabolism of enological lactic acid bacteria. So at the end of this path, I was really determined to find a PhD project during which I could study microorganisms in fermented beverages, and SpoilControl fitted perfectly with this desire. So now I am the doctoral candidate number 1 in the SpoilControl project, and my work focuses on managing spoilage potential of acetic acid bacteria in beverage industry. My work will focus on understanding which factors influence their spoilage potential, what impact they have on the beverage, how to detect them quickly, and how to design integrated control strategies. The first step is to isolate acetic acid bacteria from fermented beverages in order to build a microbial collection. And so at the moment I am cultivating acetic acid bacteria on different culture media, trying to collect as much diversity as possible. In order to improve isolation process, I am also testing the addition of antibiotics on media to reduce competition from other microbial groups. The results of these isolations will be also compared with metagenomic techniques to better understand what we are really able to isolate through culturomics compared with the whole population as shown by metagenomics. Anyway, the pure isolates obtained will be also transferred into liquid culture media, and in this way we can recover cells into their active growing phase and extract their DNA. This DNA will be used first, thanks to PCR techniques, to distinguish different acetic acid bacteria strains that we isolated in order to put them into a microbial collection. In addition, this DNA will also be used for whole genome sequencing that I will carry out at the University of Bordeaux in a few months. All this knowledge will allow us to perform the experiments I mentioned before, and so to better understand these fascinating spoilage microorganisms. This work will help the beverage industry to prevent spoilage, reducing quality losses and economical and environmental damage. At the same time, for research, it could provide new knowledge, new strains, and new tools to study these microorganisms.